Hi friends, welcome to our channel Way to Success. இந்த வீடியோவில் PGTRB Zoology Unit 1 Comparative Anatomy of Vertebrate Urinogenital Systemக்கான short notes பார்க்கலாம். Introduction. Urinogenital system is the combination of excretory and reproductive system. In the embryos, they develop independently, but in the adult, they become connected with each other. Therefore, they are usually described as urinogenital system. Kidney is the main excretory organ of vertebrates. Kidney is concerned with the elimination of metabolic waste products. It is similar in both sexes. Testis is the male reproductive organ or gonad. Ovary is the female reproductive organ or gonad. Excretory system. Waste products of metabolism include urea, ammonia, uric acid, creatinine, inorganic salts, and water. These are excreted through the kidney. Modern vertebrates exhibit three different kinds of adult kidneys: pronephros, mesonephros, and metanephros. Nephrons are the functional units of the kidney, where the blood is filtered with these metabolic waste. Metabolic waste pass to the outside through ducts. Structure and origin of a vertebrate kidney. A pair of kidneys lie dorsal to coelom in the trunk region, one on either side of dorsal aorta. Each kidney is composed of a large number of units called uriniferous tubules or nephrons. Their number, complexity, and arrangement differ in different groups of vertebrates. Kidney tubules arise in the embryo in a linear series from a special part of mesoderm called mesomere or nephrotome. It is a ribbon-like intermediate mesoderm running between segmental mesoderm and lateral plate mesoderm on either side along the entire trunk from heart to cloaca. The anterior end of the nephrotome develops into the pronephros, and the middle region mesonephros and posterior region metanephros. Nephrons are three types: pronephric tubules, mesonephric tubules, and metanephric tubules. A kidney is formed of thousands of nephrons closely packed within a connective tissue capsule. The nephron is simple in the primitive and embryonic vertebrates, but it develops complexity in the adult and higher vertebrates. A uriniferous tubule is differentiated into three parts. First one, peritoneal funnel near the free end of a uriniferous tubule, ciliated, opens into coelom via coelomostome or nephrostome for draining waste from coelomic fluid, present in embryos and larvae only. Next, second one, Malpighian body. Bowman's capsule and enclosed glomerulus together form a renal corpuscle or Malpighian body. Bowman's capsule is a blind, cup-like, hollow, double-walled sac that encloses a tuft of blood capillaries called glomerulus. A branch of renal artery called afferent glomerular arteriole supplies blood to the glomerulus. An efferent glomerular arteriole emerges out of glomerulus to join the capillary network surrounding the tubule. Third one, tubule. Malpighian bodies filter water, salts, and other substances from blood. During passage through tubules, More substances are secreted into filtrate while some are reabsorbed. All the tubules of embryonic kidney are convoluted ductules that conduct the final filtrate to a longitudinal duct which opens behind into embryonic cloaca. Types of kidneys. Kidney is simple in the lower vertebrates and complex in the higher vertebrates. There are 5 types of kidneys in vertebrates. They are archinephros, pronephros, mesonephros, opisthonephros, and metanephros. First we can see about the archinephros. It is otherwise called holonephros. It is the primitive kidney of ancestral vertebrates. It is the forerunner of vertebrate kidney, regarded as a complete kidney or holonephros, as it extended the entire length of coelom. Its tubules were segmentally arranged, one nephron for each body segment. Each tubule opened by a peritoneal funnel or nephrostome into coelom and external glomerulus. All the tubules were drained by a common longitudinal archinephric duct opening behind into cloaca. No living adult vertebrate possesses an archinephros, but kidneys of larvae of hagfish, mixin, and limbless amphibian are very much similar to archinephros. Second one, pronephros, anamniote kidney. The first kidney tubules to appear in the embryos of all vertebrates, found dorsal to the anterior end of coelom on either side, called pronephros, as they are first to appear in the embryo. Also termed head kidney due to its anterior position immediately behind the head. A pronephros consists of 3 to 15 tubules segmentally arranged. Each tubule opens into coelom by a funnel or nephrostome and also projecting into coelom near each tubule and not connected with may lie an external or naked glomerulus without capsule. Most have internal glomeruli. In some cases, glomeruli unite to form a single compound glomerulus called glomus. All tubules of pronephros open into a common pronephric duct leading posteriorly into the embryonic cloaca. It is mostly transitory and soon replaced by the next stage or mesonephros. Pronephros functional only in embryonic or larval stage of amphibians and fishes. Retained throughout the life in adult cyclostomes and a few teleost fishes, but it is non-urinary and mostly limboidal in function. Next one, mesonephros, develops from the middle part of intermediate mesoderm posterior to each pronephros soon after its degeneration in the embryo. At first, the new mesonephric tubules join the existing pronephric duct and are segmentally disposed. Later on, the tubules multiply by budding so that their segmental arrangement is disturbed due to the increased number of tubules per segment. Tubules of pronephros and mesonephros develop similarly and are homologous. However, Mesonephros is functionally better than pronephros because mesonephric tubules are more numerous, longer, and develop internal glomeruli enclosed in capsules forming Malpighian bodies. The mesonephros is also termed as Wolffian body. With the disappearance of pronephros, the old pronephric duct became the Wolffian or mesonephric duct. In amniotes, mesonephros is functional only in the embryos, replaced by metanephros in the adults. Mesonephric kidney is not metameric, but in myxoids, it is segmental and sometimes called a holonephros. Next, fourth one, ophistonephros. In sharks and caecilians, the mesonephric tubules extend posteriorly throughout the length of coelom. Such a kidney is sometimes called a posterior kidney or ophistonephros. Drained by mesonephric duct, ophistonephros is similar to mesonephros, but mesonephros appear during embryonic development in reptiles, birds, and mammals. But when it serves as an adult kidney, it is called ophistonephros. Nephrostomes are generally lacking. Renal corpuscles with internal glomeruli present. Next, the last one, metanephros, the functional kidney of higher vertebrates or amniotes. It is formed from the posterior end of the nephrogenic mesoderm, which is displaced somewhat anteriorly and laterally. When metanephric tubule develop, all the mesonephric tubules disappear except those associated with the testis in male and forming vasa efferentia. The adult kidney of amniotes differ from that of anemniotes chiefly in its origin from only caudal end of nephrogenic mesoderm. Second one, in greater multiplication and a posterior concentration of nephrons or tubules. They are particularly very large in number and highly convoluted in birds and mammals, hence the large size of kidney. It is estimated that each kidney of man is composed of about 1 million nephrons. The high rate of metabolism yields a large amount of waste to be excreted. The mammalian metanephros shows greatest organization of all, with several additional features. A thin U-shaped loop of Henle is present between proximal and distal convolutions of a metanephric tubule. Such loops are absent in reptiles and rudimentary in birds. Kidney shows an outer cortex with a concentration of renal corpuscles and an inner medulla having collecting tubules and a loop of Henle, which are aggregated into one or several pyramids tapering into pelvis. Mammalian kidneys do not receive afferent venous blood supply as there is no portal system. Thank you for watching this video.